Welcome to the Cambridge Neuroscience Interdisciplinary Seminar Series. This series features current work from across the schools and departments of the University of Cambridge, reflecting the pioneering work and diverse interests of members of Cambridge Neuroscience. Cambridge Neuroscience is currently going through quite a detailed consultation process to develop 6 new themes for the research we do here. Each of the next 12 talks will come from one of the 6 new themes, 2 from each. For more info on the themes and the talks covered in this series, please follow the links below and follow us on Twitter at CamNeuro. So today we're really delighted to welcome Professor Roger Barker. Roger is a Professor of Clinical Neuroscience here at the Department of Clinical Neurosciences and the Wellcome MRC Cambridge Stem Cell Institute. He's also a consultant neurologist at Addenbrooke's Hospital. And for those of you who don't know, his research primarily focuses on 2 neurodegenerative disorders, namely Parkinson's disease and Huntington's disease. So Roger is a great speaker, no pressure, Roger, and I'm sure you'll all really enjoy the talk. So over to you, Roger.
Well, thank you very much, Deborah, for inviting me, and thank you all very much for joining this afternoon. I don't think you're missing much on the weather, at least here in Cambridge. So what I thought I would do in this talk is it's a slight change from the title which I gave Deborah, because I'm going to talk about growth factors. I'm going to talk about cell-based therapies as a way of trying to repair the Parkinsonian brain. And I'm going to do this by walking us through a little bit of an introduction on Parkinson's disease, then a little bit on what we're hoping to do with cell therapies, what has been done with cell therapies, and where is the future of this field, which I would say is a very active field at the moment. So I just need to now move on to my next slide. So the first question, of course, is what is Parkinson's disease? And I'm sure most people in this audience will know exactly what this is, but it's useful to just understand the principles of it, because actually what we're trying to do with our therapies is slightly different to what some people will claim they're doing with their treatments. So Parkinson's disease, which takes its name from James Parkinson, who described it in 1817 in a study which would have got him struck off by the General Medical Council because there were only a few of his patients. The other were people in the street that he went and talked to and then rate them all out without any consent. But he basically described the clinical syndrome of Parkinsonism, called it the essay of the shaking palsy, was his famous piece of work on this. And Charcot in the late 19th century attributed his name to it, given his description. And amazingly, in 2021, the diagnosis of Parkinson's disease is made by someone telling you that you've got Parkinson's disease. There are no diagnostic tests. And so it's defined clinically by this resting pill rolling tremor, a slowness of movement, stiffness, and slight problems with walking with postural instability. And that clinical diagnosis is often substantiated by a scan looking at dopamine in the brain, a DAT scan, and a response to therapy. So if people have that clinical syndrome, they have an abnormal DAT scan, which relates to their dopamine terminals, so they have a loss of dopamine, and they respond well to medication. You're about 95 to 99% certain they've got Parkinson's disease. And that becomes important because there are mimics of Parkinson's disease which can create problems when you come to experimental trials. And actually, newly diagnosed Parkinson's disease patients, about 1 in 10 do not have Parkinson's disease, as it turns out. So this can create problems if you have a trial of 10 patients, because you already have a failure rate of 10% with your experimental treatment, because 1 in 10 doesn't have the condition you think they have. But for our discussion this afternoon, those are the clinical features. Pathologically, it's defined by the formation of alpha-synuclein aggregation in specific neuronal populations, particularly the midbrain dopaminergic neurons. And so pathologically, it's defined by the alpha-synuclein Lewy body and the loss of dopamine. And the number of dopamine cells you have in your midbrain, in your substantia nigra projecting up to your striatum, is about 500,000 on each side of your brain. So you have about 500,000 dopamine cells. They're not replaced during life. And when you lose half of those, about 250,000, you develop the first clinical features of Parkinson's disease. And that is important for our discussion, because replacing 250,000 dopamine cells on either side of the brain is seriously doable. Whereas trying to replace vast populations of cells in the hemisphere, say with a stroke of different types of cells, is going to be a much more challenging way forward with cell-based therapies. So we can diagnose Parkinson's disease clinically. We know what the core pathology is, but it's not the only pathology. The pathology is many other neuronal populations, not only within the brain, but outside, including in the gut. But we know that the dopamine loss, which I mentioned, is critical to the expression of the problems you see in Parkinson's disease, because all of the treatments we currently use in clinic to help people with this condition essentially target the dopaminergic network, either through inhibiting the breakdown of dopamine or directly replacing dopamine in the form of L-DOPA or dopamine agonists. And then treatments like amantadine and deep brain stimulation are used to treat the complications which arise as a consequence of long-term dopaminergic medication. So the entire treatment of Parkinson's in 2021 is around dopaminergic-based therapies and the complications therein. So that's important because that gives us a rationale for what we're going to do with our treatment. So that is what Parkinson's disease is. That's what it's clinically, that's what it's pathologically, that's how we treat it. So what are we trying to do when we talk about repairing the brain in Parkinson's disease? Well, essentially what we're trying to do is we're only trying to repair one aspect of it. So this is not a curative therapy. We are not trying to get rid of the disease. We're not going to treat pathology that lies outside that nigrostriatal pathway. But as I've already said, the response to therapy means that actually you could make a huge difference to patients if you could target that pathway in a reparative way. And for many of you who know people with Parkinson's disease in the early stages, if you give them dopaminergic medication, they essentially go back to normal. But this is what we're trying to do. We're not trying to cure it. We're not trying to deal with all the problems. We're trying to deal with the dopaminergic aspects of the disease that relate to the nigrostriatal cell loss. So how could we actually repair that network? Well, there is essentially two obvious ways in which you could do this. One is you could put in fertilizer to regrow the network that's died off. So you can add in growth factors. And that has been the subject of many trials. And some of you would have seen the BBC documentary 2 years ago on the trial of GDNS, glial cell line derived neurotrophic factor in people with Parkinson's disease, which produced a negative outcome. Patients did not improve with that growth factor in the trial, although on the television series, you could see that some patients certainly had improved. But that's not an area I'm going to talk about this afternoon. What I'm going to talk about is the notion that you're going to repair the Parkinsonian brain by replacing the lost nigral dopamine cells. And that's an important point, that I'm going to replace the dopamine cells, but I'm not going to replace them with any type of dopamine cell. The most effective therapies are those that replace the subpopulation that are lost in Parkinson's disease, which are the nigral A9 dopaminergic neurons. So anything that is a dopaminergic neuron otherwise won't be as effective. So that's what we're trying to do. We're trying to essentially have a better way of delivering dopamine to the Parkinsonian brain on the rationale that this type of therapy will work as well as the oral medication. So that's what we're trying to achieve. What's the other advantage we would have with this therapy? Why bother with it, given I've already told you we have a stack of very good therapies which exist around dopamine replacement in Parkinson's disease? Well, I would say there are primary reasons why this therapy would have an advantage over that which we can do in the clinic at the moment with our dopamine treatment, irrespective of the fact that this would be a one-off treatment and people wouldn't need to take drugs several times a day for years on end. The advantages of this approach would be if I could transplant dopaminergic neurons into the site where dopamine is lost. And in the case of Parkinson's disease, whilst the nigral is the place where we lose the cell bodies, it's the posterior putamen in the striatum where we lose the dopamine innervation. That's the area we're trying to repair with the dopamine replacement. If we could put neurons into the posterior putamen, innervate that, but dopamine levels back to normal, the advantage of that is we would be replacing dopamine where it's needed and only where it's needed. So this would avoid many of the off-target effects you see with the drugs. So many of you will be aware that the dopamine agonists through the ventral striatum can drive very abnormal behaviors, compulsive behaviors, gambling, hypersexuality, things which are very disruptive to people's lives, very destructive. You would avoid all of that. Also, the dopaminergic drugs stimulate the intact cortical system and can create people develop problems with hallucinations, paranoia, and such like. The other major advantage would be if you replaced the lost dopamine innervation with neurons releasing dopamine in a physiological way, which you would do if you put back nigral dopamine cells, then you would avoid the long-term complications which you see with the L-DOPA therapy, which is where you get this non-physiological stimulation of the dopamine receptors in the striatum, which drive the development of these so-called L-DOPA-induced dyskinesias. And these L-DOPA-induced dyskinesias in the early stages aren't a problem. People, I'm doing a little bit of it myself now, not that I'm Parkinsonian, but they tend to move their head, they jig a little bit. But after 5, 10, 15 years, those can become severely disabling. So patients actually have such violent dyskinesias, they can throw themselves onto the floor. And that's often coupled with periods where the drugs aren't working at all. So replacement of dopamine into the striatum should get rid of off periods completely and avoid them getting L-DOPA-induced dyskinesias through the use of oral non-physiological stimulation of the dopamine system with the drugs. So this therapy, it not only could be as good as dopamine drugs, but it has primary advantages over what we have in clinic at the moment, whilst not being curative. And of course, that sets up a sort of interesting philosophical discussion, if you like, about whether this is disease modifying, because people would say it's not disease modifying, because I'm not modifying the underlying disease itself, but I have significantly modified the disease as they experience it. So this, I think, is an important point to grasp, that this is a symptomatic therapy, but has the potential to change the natural history of how we treat Parkinson's disease, and could, in essence, put people back to normal for many years whilst not curing them. So we've sort of, I've tried to explain, what Parkinson's is with respect to this therapy and what we're hoping to achieve with cell-based therapies around repairing this dopaminergic network. The next question I think we have to ask ourselves, which is in fact where our work began 20 odd years ago, was who is ideal for this therapy? Now you may think, well, that's all a little bit, that's all a bit unnecessary because I've just told you about Parkinson's. But one of the important things to understand about Parkinson's disease is it is a somewhat heterogeneous disease. So if any of you have worked with patients with Parkinson's, as I'm sure many of you have, or seen and treated patients with it, it's quite clear that some patients with this condition have a lot of tremor, don't seem to progress very much. Some patients don't have any tremor, seem to be rather rigid, have major problems walking, and develop an early dementia. So it's quite clear that people follow different courses. So our original study to try and identify the optimal group for any type of therapy was really where we were starting, but particularly around dopamine cell-based therapies, was through this study called the CAMPAIGN study, which was originally done by Tom Faltney, my first clinical fellow, and then Caroline Williams-Gray, who now works up here. And we followed patients with newly diagnosed Parkinson's in the county of Cambridgeshire who were diagnosed over a 2-year period, and we followed them to the present day. And this has allowed us to come up with some fairly interesting baseline data, which, interesting, is the only community-based data has existed at the time of Parkinson's disease. So the average age you get Parkinson's is 70. If you look at mortality rate, it is no greater in the Parkinson's population compared to the age and sex-matched normal population. So at the community level, Parkinson's doesn't shorten your life. You don't tend to die of Parkinson's disease, you tend to die with it. But over the years, we've followed this cohort up. We've been very well supported by many people in the Cambridge community, Carol Brain in particular, and Trevor Robbins, who've been fantastic in helping us devise the various tasks to investigate these patients. And for our discussion this afternoon, the important thing is that by following people over time, we have come up with the idea that there are types of Parkinson's disease. They're probably not discrete types, they probably lie at the end of a spectrum. But fundamentally, there are sort of two extremes, if you like, of Parkinson's disease, as defined by that natural history study. There are people who are younger, which in our study was actually under the age of 72, and they had, interestingly, some fairly subtle cognitive deficits, which we thought related to dopamine dysfunction across the corticostriatal network. But these patients, these abnormalities are not predictive of a dementia. So these patients tend to have a disease which is very much centered on the dopaminergic system and seems to stay within that system for many years. In contrast, there are older patients over the age of 72 who at presentation couldn't list more than 20 animals in 90 seconds, couldn't draw interlocking pentagons. These were the two tests that we used. And if you were over 72 and couldn't do either of those tasks, your chances of developing a dementia at 5 years was 80 to 90-fold greater than people who were younger who could do those tasks. So whilst this could be used as a way of screening for people who are going to develop an early dementia, is probably not so much a screen, it's probably the fact that they already have the early features of it. We've just actually sought to define what that is. Now, that obviously has some implications for therapy. The question is, well, what determines whether you're in the good or the bad group outside of age, which is obviously the major factor. And this has allowed us to explore this in terms of genetic variants. So your ApoE4 genotype, whether you carry a single gene in the GBA, which is this gene which is associated with Gaucher's disease. If you have two abnormal genes for that, you get Gaucher's. If you have one, it greatly increases your risk of Parkinson's disease, and it greatly increases your risk of developing an early dementia. There's a new gene which is about to be published from a collaboration we've been involved with people in the States around a synaptic a gene which codes for a synaptic protein, RIMS2, and then the tau haplotype. And Caroline has also demonstrated that levels of peripheral inflammation are predictive of getting an early dementia. So this type of work has enabled us to sort of start to understand why patients follow different courses. That obviously has implications in terms of developing predictive models. So we can now put some of these factors into models and work out who's going to do well and who's going to do badly. It gives us the scope for exploring the biological basis for this, either through genetic studies or actually growing cells from the patients and studying pathology within them. But for our discussion this afternoon, it has therapeutic implications. In other words, what we don't want to do is we don't want to take an experimental therapy, especially a cell-based therapy, injecting it in someone's head. You're not going to be able to do that in vast numbers of patients. But you want to make sure that the group you're going to start with are the group who are likely to benefit from it. And so that's obviously the group who are the younger patients who have this more dopaminergic centric therapy versus the patients who seem to have a more malignant disease who might be better suited for disease modifying therapies. Now, just to sort of illustrate this point, if we don't take this into account, and let's say we do a trial of 8 patients, and we happen by chance to take 4 from the good group with the dopaminergic centric disease and 4 who are the older patients with the mild cognitive impairment, we transplant in dopamine cells. We wait 3 years because that's a good time to wait to see if the cell's going to work. At the end of that 3-year period, we ask the patients, do you feel better for your cell-based therapy? Something that NICE would very much like, a very subjective but quality of life, if you like, for the patients. The 4 who've got the younger onset disease will say, Roger, well, that's pretty crackerjack. I feel pretty good with that therapy. It seems to have made a big difference. The other 4 patients have started developing more florid features of dementia, and they'll say, Roger, I am actually no better for this therapy. To be quite honest, I'm worse. I'm now hallucinating. I'm becoming paranoid. So you take 4 patients who've done rather well, 4 patients who've done rather badly, you combine them together, and you think, actually, it makes no difference. And so whilst that's a very sort of superficial way of sort of illustrating the point, this has been a major problem in the field of reparative therapies for Parkinson's, that not enough attention has gone in to selecting these patients. So about 10 or 11 years ago, we took this epidemiological data into a study which was funded through the European Union called TRANSEURO, which is a study which involved 2 major elements. One was a natural history study where we collected younger patients who we thought would fall within this good prognostic group and follow them over time to validate what we suggested at the community level. And out of this population, the second study was to be grafting in dopamine cells to repair the brain in that patient cohort on the grounds that they would be the optimal group for doing this. So it was a sort of nested study, if you like. We collected 150 patients from multiple sites across Europe, and we would randomly select, the idea was 20 patients out of the 150 and graft them, and then compare the 20 grafted patients against the 130 that did not have the transplants without having to resort to sham surgery and other such interventions, which has become a major topic in this whole area. So I'm not going to go into huge detail with this, but this was essentially a paper we published a couple of years ago where we analysed this data and some of the lessons we'd learned en route with setting up this study for the transplant study. And essentially, we used these younger patients who are earlier in the disease course. The details of this are not that critical, and we collected patients from a number of sites, Cambridge being the major site, as well as University College in London, Imperial in London, Lund in Sweden, who we work with very closely, and then other centres, one in Cardiff, one in Paris, and one in Freiburg. And essentially what we demonstrated was that over the first 36 months we collected this data, everything seemed to fit in with what we'd predicted. In other words, the change, so these are things called the Unified Parkinson's Rating Scale. Part 3 is the motor score, and of elements of that, some are more responsive to dopamine drugs than others. So we sort of stratified our ratings based on that. And you can see across the graphs at the top that over a 36-month period, actually patients progress in a rather linear fashion, in a rather satisfying way over that period of time, so that if we were to intervene, there would be enough change for us to be able to see it, and it would be fairly predictable. And none of the patients, although it's obviously the average, disappeared off the graphs in directions we weren't expecting. So the population seemed to be optimal for what we were planning to do. And on the bottom, we have the Addenbrooke's cognitive examination, which many of you will know is scored out of 100. So these patients over a 3-year period went from about 96 to 97, otherwise they were entirely normal. So no cognitive deficits, which is what we predicted. And the AIMS is an abnormal involuntary movement scale, which again is very low scores, showing that none of these patients developed dyskinesias. So this population very much fitted in with what we predicted to be. So we now think that we've identified the group who would be optimal for an intervention around a dopaminergic based therapy. And so then the question arises as to what that therapy will be. And so today I'm going to be talking about cell-based therapies and where we've got to with respect to that. So the first question, you've now defined your population, you've defined your rationale. So I'm going for this group of patients with Parkinson's who seem to have a largely dopaminergic nigrostriatal pathology. They respond well to medication. They don't have any other complications. I'm going to replace their dopaminergic system with a cell-based transplant. So then the question is, well, what dopamine cell would you like to put in? And almost anything you can think of has been tried in a trial somewhere in the world, for which the basis of many of these is interesting reading. So one thing you could start with, you could say, which is where a lot of this field began in the 1980s, well, why don't we just take a dopamine cell from the patient in some other site and just whip it out and shove it in their head? Now, obviously, the first question is, well, where else do you make dopamine in your body? And there aren't many places you make dopamine. So the adrenal medulla, obviously the adrenal gland which sits next to the kidney, produces minuscule amounts of dopamine, but people were convinced that that would be enough. The carotid body, which obviously sits next to your carotid artery, has also got dopamine in it. So people thought that might be quite a sensible place to start by just putting dopamine cells from one place in your body to another, autologous grafting. The alternative would be to take dopamine cells from the developing aborted fetus, so take out the developing midbrain, the ventral mesencephalic area, and put that into the brains of people with Parkinson's disease. So take the developing dopamine cells from human fetuses, an allogeneic transplant. A third option, which only really became possible in the late 1990s, when the first human embryonic stem cell lines were made by Jamie Thompson, is to take embryonic stem cell lines made from spare embryos in IVF programs, differentiate them down to dopamine cells, and then transplant those into the brain of patients with Parkinson's disease. That would be allogeneic, because it would obviously be from one unrelated human to another. And then the final, which only became possible in 2007, really, when Shinya Yamanaka made iPS cells, induced pluripotent stem cells from adult somatic cells, normally skin or blood, and you turn those through an iPS intermediary into a dopamine cell, which again, for most people, that would be an allogeneic. It would be from an unrelated person who would be providing the iPS. But obviously, this gives you the potential to do autologous grafting. You could take the patient's own cells, turn them into iPS cells, turn them into dopamine cells, and pop them back into their brain. So where have we got to with all of this? What has actually worked? Well, I would say the first thing is that all of those have been a bit of a disaster. So during the 80s and 90s, these adult cells were used. There was a paper in the New England Journal in 1987 from a Mexican group showing that adrenal transplants transformed patients with Parkinson's, which led to hundreds of similar transplants being done in America before someone audited the data and said it didn't actually work. The cells didn't survive and it had a significant morbidity. So the idea of doing autologous grafting using the patient's own cells, own adult cells which produce dopamine, is no longer a viable option, although autologous, as I say, has now been considered around iPS cells. The most successful to date, and for which the most data is there, are the fetal dopamine cells collected from aborted human fetuses. And so this has been going on since the late 1980s. And what we can say about this approach, so the idea here is that under proper ethical governance, you collect, women donate their fetal tissue for research. They can't stipulate what research. That is then collected. You dissect out the developing midbrain from those aborted fetuses, you turn it into a crude cell suspension, and then you inject it into the brain, into the striatum, into the posterior putamen of patients with Parkinson's disease. This was first done in '88 and carried on through the latter part of the 20th century. And recently we've just completed a trial with this, although we don't have the data to present yet. But essentially from these early trials, what you can deduce is that this approach can have long-term clinical benefits. So proof of principle, this can work. So you take fetal dopamine cells from several fetuses. You need at least 3 per side of the brain to make enough dopamine cells. If you put them into the Parkinsonian brain, then people will have surviving transplants long-term with functional benefits. Now, this is very difficult to see, but essentially the UPDRS motor score up on the y-axis over time, you could see in these 2 patients that over a 20-year period from the time of grafting, their motor scores have not changed. So their scores are around 20, which is probably less than they had when they first presented to their neurologist 10 years before they had this transplant. So 30 years after developing Parkinson's disease, these patients with these fetal dopamine transplants have motor scores which are less than when they presented. They're on no medication. And when you look at their dopamine scans in their brain, the areas which have been grafted have now been returned back to normal in terms of dopamine levels. So this shows that in some patients, these were only 2 patients out of 18 treated in Lund in Sweden, that they were restored to normal in terms of their clinical and imaging modalities. It's also been shown that these cells survive long term, which is what you'd expect based on the PET imaging, which is obviously looking at dopamine terminals. So this was a patient who was grafted in the late 1980s, patient 4 in the Lund series, who died 24 years later in 2013, only had a transplant on one side of his brain. And when they looked at his brain 24 years after the grafting, next to the sort of schematic of the brain, the white rectangle is the non-grafted side, and everything you see on that picture, which is brown, are transplanted dopamine cells which have survived a quarter of a century in the Parkinsonian brain. Now, some of those cells did develop the pathology of Parkinson's disease, which is an interesting story in its own right, but the important thing is that the cells have functional benefit both clinically and on imaging, and that correlates with what you find at post-mortem. So these transplants can survive and have a functional benefit. By virtue of the fact you have improved motor control, it also has effects on non-motor aspects of the disease. So if you've got better motor control, you can turn over better in bed at night. So you sleep better. If you sleep better, you think better, you feel better. So non-motor and quality of life aspects were improved by this therapy, which, as I said at the beginning, is not curative, but symptomatic. And also this transplant was shown to release dopamine in a physiological way. So this PET study, which was done in '99, was done on the patient who died in 2013. So the patient's post-mortem brain you're looking at, this is what his brain looked like in 1999 when he was imaged at the Hammersmith. And in this study, what it was demonstrated was that if you stimulated the transplant with amphetamine and looked at dopamine release by using a PET imaging technique that looked at raclopride displacement, then the patient's transplant released dopamine in the same way as the intact striatum of people without Parkinson's disease. So it seemed that this actually survived long-term, these cells, their functional benefit, they improve many aspects of Parkinson's disease, and they release dopamine in a physiological way. So then the question is, well, why, if this is such wonderful stuff, why is it we're not doing this all the time, and why isn't this a more mainline therapy? Well, there are 2 reasons for that. One is obviously the ethics around this is complicated because this is human fetal material from aborted fetuses. It's also because there were 2 trials which ran out of the Clinton administration in the sense that he allowed federal funding for fetal tissue, which had not been allowed under Bush Senior before that, which enabled NIH to fund 2 trials of this approach, neither of which actually produced positive outcomes for a number of reasons. As a result of which, by the turn of the century, this therapy, by virtue of these two double-blind placebo-controlled trials failing to hit their primary endpoint, had largely gone out of fashion as deep brain stimulation came very much into fashion. Now, of course, you could say, well, that's perfectly reasonable. This therapy doesn't work. The more formal trials didn't show that it worked. But you can't get away from the fact that I've also shown you a couple of patients where things have clearly gone in a very different direction. So we attempted about 10 years ago, when TRANSEURO first came into existence, to do a meta-analysis of what data we had on the trials. And it was very difficult to do this. The numbers are very small. And essentially what we tried to do was we tried to identify what we thought were the key factors which seem to encourage a good response to the therapy. So on this particular graph, this is a greater than 33% improvement in the UPDRS, which is, I say, the standard motor measure, 2 years after having a transplant. And about a third of all the patients in these major studies have shown such a response. And what characterized those patients were that they were younger, less advanced patients, exactly as we would have predicted from our epidemiological study, and as we've already shown with our TRANSEURO observational study. That there were issues around, actually, they had the tissue prepared in a better way. So actually there was probably greater survival of dopamine cells with better implantation of it. They actually had immunosuppression, which some of the trials didn't give the patient. So even though it was allogeneic tissue and it's put in the brain and so it's immunologically privileged, some of them didn't have immunosuppression, but patients who were in receipt of that seemed to do better. And follow-up seemed to be a critical factor in that, obviously, if you put dopamine cells into people's brains, they will take a time to survive, mature, and then you have to some extent, learn how to use them. So if you go for a 6 month to 12-month follow-up, you will miss the major benefit of your transplant, which was the case in some of these trials. So on that background, we then set up the transplant trial of TRANSEURO on the back of the observational study that I've mentioned. So we selected our optimal patient cohort, we standardized how to prepare the tissue, we optimized how to deliver it, and then we grafted the patients. Which started in 2015, finished in 2018, and we've just reached the last 3-year endpoint on our last patient. Now, for those of you who think this is a relatively straightforward process, it obviously looks quite straightforward, but I will now illustrate some of the problems we had en route in conducting this trial. So the point here is not to put people off, but just to tell you that you have to get into the long haul in order to understand just how much work this involves. But there is an important point I'll get to at the end. So this trial was supposed to have started in 2012. It started in 2015. In the original grant, there was supposed to be 2 trials, one of 20 and one of 60. We ended up doing 10.5 patients over an 8-year period. So why did it take us so long? We had problems with the MHRA deciding whether we had an advanced therapy or not. Trying to do things across Europe proved difficult because different countries saw the trial differently. We had this was a European grant, so we had a small medium enterprise that suddenly came into existence for the imaging, which didn't exist when we started. There were new documents required by the regulatory authorities. We had a management company to help us run this that went bankrupt. We had a work package leader who sadly died of glioma, which put a major hole in the program. We had another PI who left under slightly dubious circumstances from where he was working with a whole stack of data. We had problems in Sweden with permits. We had things in the GMP lab where we prepared the tissue which broke down unexpectedly. We had problems in Sweden of working out if we had a room where we could do this. We suddenly discovered that we couldn't reproducibly get the fetal tissue into the suspension we wanted. The hospital here suddenly changed the insurance, so we couldn't use the device we were planning to use on the eve of the first transplant. The hospital moved to the EPIC electronic hospital system, so all elective surgery was cancelled, including our trials. We couldn't get the instrument sterilized properly in the hospital, and when we did, it didn't work. They decided during all of this to rebuild neurosurgical theatres, so we couldn't use that. Then the junior doctors went on strike, so we couldn't get any patients in to actually clerk them in. By which time all the reagents, which we'd spent a fortune getting, had passed their validation date, so we had to get more made. We had problems getting tissue shipped from one site to another. We then left the EU, which has of course made it very difficult for us to do things across different countries. And now with COVID, we've also had problems following up these patients. Apart from that, it went like an absolute dream. But the important point I want to end up with is that regardless of the outcome of this trial, it is a non-viable way forward because of the 21 surgeries that we did. One patient had only half of their brain done. We had another 87 surgeries cancelled because we had insufficient fetal material. So if I'd had a stem cell dopamine cell at the beginning of this program, apart from the fact I could have started much earlier, I could have grafted in the time it took me to do 10.5 patients with fetal tissue. In theory, I could have done 108 patients with my stem cell therapy. So that is why we need to move towards a stem cell therapy. So what are the advantages and disadvantages, and where have we got to with respect to this? Well, obviously, as I've said, with induced pluripotent stem cells, you have the potential to do autologous grafting. So you, as I say, take the patient's own cells, turn them into dopamine cells, put them in the brain, which of course is ethically much more neutral. It obviously gets around problems of rejection if you believe that the iPS cells are not immunogenic, which in itself is still controversial. But in theory, you could just put the patient's own cells back, no practical problems, no immunological problems, no ethical problems. The difficulty with that is, of course, the patient developed Parkinson's in the first place. So you're putting back your engineered cells, which probably have a higher risk of developing pathology because they come from the patient. And the difficulty at the moment is the regulatory pathway for doing that. If I was to give myself a stem cell therapy, which of course would be illegal, but if I did, and Dervala a stem cell therapy from our own skin cells, for each of us, it would currently cost around £4 million to develop that therapy and test it. So for most people, this is now using allogeneic, and in Japan, they're trying to develop banks of cells so you can at least match to some extent. ES cells obviously have been around much longer. They're in clinical trials. They've been used extensively in some other diseases. There are ethical problems with their derivation. You have to destroy the embryo in order to make the embryonic stem cell line. So that creates problems in certain countries. And also, as we discovered in our trial, that where you make or where you get your embryonic stem cell can have a problem. So if you have an embryonic stem cell that is made in a country that has had new variant CJD, then you cannot use it in America for risk that you might be giving new variant CJD to your patients. You may think, well, I don't care because I don't want to do a trial in America. I just want to do it in Cambridge. If you ever want this to fly, you're going to need a commercial partner. And if a commercial partner does not have the American market, then you are going to struggle. So just to give you some idea how this field has taken off, when we grafted our first patient in TRANSEURO fetal tissue in 2015, the world investment in cell-based therapies for Parkinson's was approximately $0. By the time we grafted our last patient in 2018, it was approximately $1+ billion. So this field has become a major industry. So in terms of embryonic stem cells, we have worked with Lund with our own embryonic stem cell derived dopamine cell, which will be going to trial next year, and which we spend the most enormous amount of time working on. I'll come back to that. There's a group in New York led by Lorenz Studi and Vivian Tabar, who have an embryonic stem cell trial that's just got FDA approval for a clinical trial. They set up a company called Blue Rock, which is now owned by Bayer Pharmaceuticals. In terms of iPS cells, the Japanese group have developed this therapy. Obviously, that's where it was originally discovered or made, the first iPS cells. They have a company attached to them. Fujifilm Cellular Dynamics and Sana Therapeutics are now working on an iPS-derived therapy in Chicago. Aspen Neuroscience are using an autologous approach, and they're a group in Harvard and Mass General who have also got a program of autologous grafting. So in terms of putting this on a map, we work now with Novo Nordisk, the fourth biggest pharma company on the planet, to develop dopamine therapies for people with Parkinson's disease. I've discussed the group in New York, the group in Chicago, the group in California, the group in Kyoto in Japan. There's also Chinese trials done with both ES cells and these pathogenetic embryonic stem cells, which are non-fertilized eggs, which get around some of the ethical problems but create all sorts of other issues. And then there's a trial going on in Melbourne with a pathogenetic stem cell therapy, which has yet to report. And in order to try and avoid some of the problems we had in the fetal tissue field, we decided that it would be important to try and come together as academic groups to work together. So this is GFORCE PD when we last met in person in Cambridge 2 years ago. This has enabled us to sort of try and rationalize and learn from each other as to how we can best do this and coordinate our activities. And this has been a very positive outcome of this group that we meet every year. We had our last meeting a couple of months ago to discuss exactly where we are with all of our unpublished data. In terms of exactly where this has got to, in my sort of concluding comments, in human iPS cells, the group in Japan led by Jun Takahashi had this paper in Nature 4 years ago, showing that if you made iPS cells from patients or from non-affected patients, transplanted them into non-human primates, the cells survived for up to 2 years with functional benefits up to a year once the cells had matured. This then has led them to undertake allogeneic iPS transplant trial. The first patient was grafted in November of 2018 on one side of the brain. The other side was done in June of 2019, and they have now grafted 5 patients with iPS-derived dopamine cells. So that trial is ongoing. They're planning to graft 8 patients, and we will be interested to see what their data shows. There was this paper that appeared last year, which is a very, very controversial for all sorts of reasons, but I'm just going to gloss over that and just tell you about how some papers gain a prominence, which perhaps they don't necessarily merit based on the data. So this was a brief report in the New England Journal of Medicine, as many of you know, top medical journal in the world, impact factor of 50 odd. So this was a single case of a patient where they took their own skin cells, turned them into dopamine cells, and grafted them. Their preclinical data looked suboptimal. The cells did not look as well and as good as the other protocols which exist. On imaging, there was no change subsequent to the transplant. In terms of their objective motor assessment, the UPDRS, there was no change. So this transplant didn't work clinically, and that would correlate with its imaging. But in terms of their quality of life, their PDQ-39, they thought they'd done extremely well. They went from a very high pre-operative disability to normality afterwards, which may have something to do with how this trial was funded, which I'm happy to take in questions, but I'll say nothing more at the moment. In terms of our own program of work, and that in New York, the group in Lund and in New York have produced protocols which enable you to reliably produce large numbers of dopaminergic cells of the A9 subtype, which is what I said at the beginning. And one of the reasons this has caught fire is that it's highly reproducible and it's highly efficient. So here we have a graph showing reduction in drug-induced rotation with 3 different cell lines, the H9, the RC17, which is the one we're going to clinical trial, and a new line from the Karolinska, different batches, some cryopreserved. They all reverse drug-induced rotation, a rather crude test, but nevertheless a very reliable test of dopamine content within your transplant. And the important point is that starting manually with a 6-well plate in your GMP facility, so if I have the skills, which I don't, but if someone went into a GMP facility with a 6-well plate of ES cells, then over a 16-day period, they would produce enough dopamine precursors to treat 500 patients. So if you scale that up to 1,000, then you obviously could do 500,000 patients, which is more than exist in Europe with Parkinson's disease, or would be suitable for this type of therapy. So it suddenly becomes economically very viable, this particular therapy. So that is why I think to some extent it's caught fire. It's very efficient to make it. It's very reproducible. It's very reliable. Lots of labs can do it. We now have predictive markers, so we know when we've made the cells whether they're likely to turn into good mature A9 dopaminergic neurons, and they seem to have a potency which is similar to that which you see with fetal tissues. So they look to be the real thing. So that has enabled us to start planning for our trial, which, as I say, we've been doing for a year and a bit, and we plan to go to trial next year. And the group in New York are about to start delivering that. Obviously, there's huge amounts of work you need to do in terms of quality control testing of the stem cell line and the stem cell product. You have to do a vast amount of animals as regulated by the MHRA. So these are of good laboratory practice. So if you don't know what that is, just do what you do in your lab and add 3 noughts to what it costs you, and that will be GLP. Incredibly expensive. Efficacy studies we've done in our own labs, and all of that data is now being collected to allow us to actually look at the data, all of which looks fine. The cells look safe. They look as effective as we would expect them to be. We've also looked at their immunogenicity as a way of trying to say whether our immune therapy of the patients who receive this would be the same as we do for our fetal transplants as we did in TRANSEURO. As with fetal tissue, these cells don't really express much in the way of MHC class 1 antigens, HLA class 1. They do if you stimulate them with cytokines, which would be the situation when you put them in the brain. We also discovered that these cells are not particularly do not do well with certain immunosuppressive therapies, which means especially mycophenolate, which is what we were going to use. So we've actually had to change our immunotherapy as planned. And we're now trying to move this into in vivo studies. Finally, we obviously have to solve the problem of delivery device, given the problems we have in TRANSEURO. This is not a trivial exercise. You have to be able to have a device which can make sure your cells survive, deliver the cells well across the target area, don't have any problems with the device itself, and ideally doesn't require intraoperative MRI, which is what most of the other centres are opting for, which would greatly restrict your ability to run this therapy out across multiple centres. For example, Cambridge does not have intraoperative MRI. So if this was the only way you could deliver this therapy, we could not do this in Cambridge, like most neurosurgical centres. The device which was used in the TRANSEURO program has been varied. This is an MRI scan from the neurosurgeon in Lund, and all those black holes are where the cells were deposited. So with the right device, you get a beautiful delivery of the cells. But this is not a trivial exercise, and is currently costing several £1 million to develop a new device for delivering our cells for this trial. So where are we at the moment with this? Well, we're obviously working, as I say, towards our first in-human study, which will be next year, where we will have 4 patients, 2 here in Cambridge, 2 in Lund at dose 1, and we will then decide whether we will escalate to a second dose and 4 more patients, 2 here and 2 in Lund. We may opt to stay for dose 1. So this is in some ways dose finding, but the first dose we think will be sufficient to give patients enough dopamine cells to have a major functional benefit. So just to sort of conclude, this has been a long program of work starting in 2000, where we set about trying to identify our optimal patient group, which we think we've done. These younger patients with Parkinson's disease earlier in the disease course without significant L-DOPA-induced dyskinesias. We've identified a stem cell source, which we think we can take globally, even though I've said there are problems with taking a UK line into America. We think we can get exemption because it's for more elderly people and it's a single use into the brain. We have done all the necessary in vitro and in vivo studies with research grade protocols to show that we can make the cells as we see them and we can get the functional efficacy which we need. That's not good enough. You have to do that to GMP without it altering the product, and that is what we've demonstrated. You have to do the preclinical testing, which costs, in our particular case, that costs about £1 million, which is just showing that the product is safe and it's efficacious, and that has now been completed. We're still trying to work out the best way to deliver it. We've planned our first in-human study and we'll be submitting the application soon. And then the main purpose of that study is simply to show that it's feasible and tolerable. That's all you can do in these phase 1, phase 2A studies, but obviously we want to see sub-signal of proof of principle on imaging and clinically, and that will then open up the possibility of taking this all the way to market with the help of Novo Nordisk and their follow-up studies, which are the TRANSCEND studies. So just to conclude, I would say that, importantly for our discussion today, how can we fix the brain in Parkinson's with cell-based therapies? Not all patients are going to be suitable for it, but there will be some who clearly will. It's not curative, but it does have the potential to dramatically change what we can do for Parkinson's. And if it worked, then everything we do in Parkinson's today would be redundant. So everything we do, including deep brain stimulation, would not be necessary, except possibly for trauma-dominant patients. The protocols exist, and there's now clearly clinical trials are coming forward with this. And so in the next few years, these trials will publish, but they need to be looked at very critically based on what we've seen, for example, with that New England medicine paper. And our own trial, we're hoping to start next year with this cell line derived from the Roslin cell ES line in Scotland. And finally, one of the joys of doing this work is, apart from the journey you take and the exciting experimental nature of it, is the people that you work with. So fabulous people that I've had in my group over the years, fabulous people in Cambridge who were very helpful, especially when I started, Carol and Trevor in particular. The wonderful team in Lund, who I work with very closely, especially Marlin Parmnagneta, who came up with the protocols, Anders Bjorklund, who basically invented cell-based therapies for the brain, and then these wonderful consortia we've had, TRANSEURO, GFORCE PD, and more recently a new European thing called neurostem cell reconstruct. And with that, I'll finish and be very happy to take any questions.
Thank you, Roger, for that really wonderful talk. Join us next week when we welcome Dr. Lucy Check from the Department of Psychology, where she will talk to us on the very timely COVID and cognition. Lucy is head of the Cognition and Motivated Behavior Lab, and her research explores various aspects of learning and memory, and in the past few years has focused particularly on the association between memory and obesity. For more on this seminar series and all things neuroscience related here in Cambridge, follow us on Twitter at CamNeuro and follow the links below. See you all next time.